Biology topic 3.1 Genes.7. The entire base sequence of human genes was sequenced by the Human Genome Project. First, we'll be looking at a timeline of the Human Genome Project, which was put together to try and sequence the human genome. In 1990, we had the start of the project, and the aim was to find the base sequence of the human genome. In the year 2000, we saw rapid improvements in the base sequencing techniques, and subsequently there was draft sequence that was published. And then in the year 2003, there was a complete sequence that was published. Although, unfortunately, not everything made sense. At least in the beginning. Although, one of the things that they were able to find in 2003 was that it was possible to predict which base sequences were protein coding. And these were genes, which there were approximately 23,000 of. So in all of this, even though we didn't necessarily understand what the entire sequence was, of our DNA at least, we were able to, in this case, predict what certain parts of this genome did, maybe how they functioned, and especially which of them were protein coding, so meaning which of the segments of the DNA that we were able to literally read and understand coded protein. Another thing that was learned was that not all of the genes were transcribed, and a lot of these were referred to as junk DNA. And this junk DNA essentially is DNA that either we didn't understand, or we still do not understand what it does, or we expect that it does nothing. And today we think that junk regions include elements that affect gene expression. And what this means is that we expect that part of the DNA in these regions of what we think of as junk DNA are there to somehow affect the gene expression, meaning, for example, one of your genes, let's say, again, the color of your eyes, the way it is expressed is the way that the gene is shown to work in you as a human. So it's not necessarily your body that is expressing the gene, but the gene itself is being expressed in a certain way because it's reading your DNA, and from the DNA knowing how to function or how to be expressed. And so we think that parts of this junk DNA affects how certain genes are expressed. Another thing we see when looking at DNA is what we call satellite DNA. And satellite DNA are simply highly repetitive sequences. The final part of point 7 is this difference between a genome versus the genome. And what's important to realize here is that the Human Genome Project, they sequenced a genome. They took a person and their genome, not the genome of all humans. So even though most of the genome is the same across all humans, at least in the case of a human genome, there are differences, and the differences are found in the single nucleotide polymorphisms, so in SNPs, or rather SNPs, which are pronounced SNPs. And this is important as these small differences are what create, or rather what lead to, human diversity. And then one final note is that we've also sequenced lots of animal DNA as well to see whether there are similarities or not only between humans and animals, but also animals in themselves, to try and get a better idea of how evolution went forward, and to be able to draw some of those evolutionary trees that you might see with all of the different kingdoms, and then all of the different steps along evolution that we see, for example, that humans originated however many years ago, and before that there was something, and then something before that, and then originally there's some original ancestor, or mutual ancestor, and we see all of this simply, well, in part by how DNA changes depending on the species and over time.